Okay, thank you very much for kind introduction. And as you all know, NK cells are cytotoxic innate lymphoid cells responsible for the elimination of infected or transformed cells by direct killing. And at the same time, they produce a lot of cytokine modulation. And they express panels of receptors, so their target recognition is extremely complicated. So they express panels of inhibitory receptors and also a lot of activating receptors. And the people claim that they somehow integrate these receptors to make their decision. One of the most known example is missing self hypothesis. So cancer cells tend to downregulate class 1 MHC molecules to escape T-cell mediated killing. And then in that case, NK cells recognize the absence of HLA or class 1 MHC using their killer molecules and kill the target cells. So they don't do bad things for normal cells, but they can kill MHC class 1 downregulated cancer cells. And also cancer cells or stressed cells express stress-induced ligands such as MIG-A that can be recognized by activating receptors such as NKG2D, so they can kill some abnormal cells. And at the same time, this kind of cure HLA mismatching can be useful for allogeneic reaction. But how these signals are integrated, that's very complicated. So Jordan Orange actually made a nice cartoon showing there are 16 steps of NK cells making their decision for killing. That means they are very conservative and very careful. But their decision-making step is very, very slow in some sense, and also inhibitory signal can get into this any of the stage to override this. So. And I don't think we can do those kind of 16-step classification. So we somehow simplified it and make 5 distinct steps. So we can directly visualize their interaction with the target cells. And we can also label the lytic granule, which is the essential part for the killing. Using the lysot sensor or lysot tracker and look at their dynamics. So they can make transient interaction at the beginning, and at some time they polarize their converge the lytic granule to the empty OC, and then they form stable synapse. And then those lytic granules are polarized toward the synapse, and then exocytosis occur to kill the target cells. So that's how we classify them. And in step, let me show you some of their examples. So we have NK cells and we have target cells coming next to them. And in this case, they somehow don't recognize it as a cancer cell, so just touch it and make only a transient contact. And later, but in some cases, they can recognize, but they don't kill. So this NK cell make stable synapse. So that's a good thing, but the bad thing is their granules are still on the backward in the distal pole. If you see that carefully, they make synapse, but the granules are still the backward side. So they are very, very careful. But when they become confident about everything, they just undergo exocytosis of granules and kill the target cells. So that it's really stepwise and conservative process. And we do this kind of live cell imaging to identify what NK cells are thinking and how they make decisions. So the first part that I want to talk is NOGO receptor, role of NOGO receptor 1. So NOGO receptor 1 is very well characterized receptor in neurobiology. And it signals through RhoA and modulates cytoactin dynamics. And their function in neuroscience is known as axon growth and synapse function inhibitor by regulating this actin dynamics. And they are also expressed in monocytes and T cells. And their function is characterized in the context of neuroinflammation, but their function in NK cells are not known. And my collaborator, Dr. Taedon Kim in CRIP, actually he was a neuroscientist in training, but he got into NK cell field and he became interested in this receptor. So he checked whether NK cells are expressing NOGO receptor 1, and he found cord blood NK, peripheral blood NK from human, or mouse NK, or NK92 cells, all express NOGO receptor 1. And he made a knockout mouse and challenged the mouse with B16 by tail vein injection to let them undergo lung metastasis, and compared the effect and found that actually knockout mouse there are much less metastasis going. That indicates that NOGO receptor 1 works as NK inhibitory receptor. And afterward, he asked me to further characterize the dynamics. So we collaborated, and actually their signaling pathway was not that different from what people know from the neurological system. So it works through ROA, ROCK, and the limb kinase, and then phosphorylation of coffulin to regulate actin dynamics. So when we look at we express the F-actin GFP into the NK92 cells to look at actin dynamics. So in the absence of NOGO receptor 1 signal, you see a relatively resting cell with a low F-actin intensity. But when we treated them with the agonistic peptide for NOGO receptor 1, we see multiple protrusion, random protrusion formation with increased F-actin intensity. Then we looked at their interaction, how they interact or regulate the cell-cell interaction. So NK cells and glioblastoma cells are in white boundary, and they express highly express NOGO A, which is ligand for NOGO receptor 1. And you see that they kind of only transiently interact and then detach and move to another cell, and so on and so forth. But when we block NOGO receptor 1 using the peptide, they can make a very stable interaction and finally, ultimately kill the tumor cells. And we look at the actin dynamics, and again, in the absence of NOGO receptor 1 blocking, they made transient interaction, and then even though they are contacting the cancer cells, they generate actin protrusion outward to the synapse direction to detach from it. But when we block it, we can see stable polarization of F-actin toward the tumor cells. So this indicates that this NOGO receptor 1 actually determine the interaction between target cells and the NK cells in cytoskeleton levels. So it's very fast and upstream signal efficiently let the NK cells move to other cells. So this is very useful in terms of immunosurveillance by allowing NK cells to scan many different cells, but that's bad for cancer cell recognition. And the second part, I'm going to talk about the role of extracellular adhesion. So cell-cell contact occur in the context of 3D microenvironment where extracellular adhesion molecules are ubiquitously presenting. And as I mentioned, our lab always look at cell-cell interaction by microscopy. And when we mix the gamma delta cells with blood cancer cells, they just form this kind of large aggregate. And some cells are dying, but we don't know which cell is which. And always this kind of problem always makes us an opportunity to publish another paper. So we decided to make this kind of single cell array of blood cancer cells. We made a single cell array of blood cancer cells, and now NK cells can engage one cell at one time and then migrate to another cell after killing or after not killing. And we coated the background side either with fibronectin or BSA. So fibronectin is very typical cell adhesion molecule in ECM, and BSA blocks integrin-mediated adhesion. And in the these red dots are cancer cells, and when we look at the BSA coated microware, NK cells are making very, very stable interaction. But fibronectin, they are really distracted by the signal of fibronectin, so they are really dancing, but not really focusing. And we measured two different parameters. One is engagement to killing, how fast they kill the target. But the second thing is the detachment time. So after killing, they have to detach and migrate to another target. And T1, so when we compared the fibronectin and the BSA case, it's very straightforward that the BSA1 did much better job. They killed much faster. But when we look at the detachment, they should take much longer because it's very sleepy surface, nothing to grab afterward. So when we compare the total time for killing, they were comparable. And when we make the cancer cell density higher by making this single cell array closer, now something different happened. So with BSA, again, they don't care whether the neighboring cancer cells are close, but with the fibronectin, hit one guy and then engage another and hit another guy, and it's really moving back and forth. And when we assess the serial killing, this one mainly kill one or at most two, but in this case, they kill up to three cancer cells. So this adhesion, extracellular adhesion, really distracting NK cells, but distraction is not always bad. They can also look at outside and do much other things. And for solid cancer cells, we made different types of microwaves. So now we can put cancer cells and NK cells in the same way as boxing matching. And when we use the BSA coated microware, the cancer cells also cannot adhere, so they become round shape, and then NK cells engage and fight. And with this fibronectin coating, cancer cells spread and NK cells engage and kill. So when we observe their interaction, BSA, this rounded tumor cells and the NK cells, very stable synapse formation and killing as blood cancer cells. But with fibronectin, cancer cells are dynamic, but NK cells are even more dynamic. They just migrate crazily. And the killing time was much faster with the BSA cases, so that's nothing different. But when we compared the NK cells with the T cells, so we used B16F10 OVA and OT1 mouse, and the BSA, again, no adhesion of cancer cells, and the OT1 engage, and then make a stable synapse and kill. But with fibronectin, the response was different. Even though the existence of fibronectin, OT1 really focused on these cancer cells and completely kill it. And this and also, even so, the mode of motility of NK cell may not be matter because the faster killing occurred with BSA. So that this result indicates that the adhesion state, so the fact that solid tumor cells are adhering on the substrate, that make it more resistant to immune cell mediated killing. And it also reminded me some intravital imaging that I show from Philip Busso's work. So he found NK cells when they are in solid tumor microenvironment, they are continuously moving, but they kill tumor cells very well. In contrast, T cells are making really stable interaction, and they didn't understand why, but I think that's because of the adhesion microenvironment, because NK cells are more distracting by the adhesion signals. So let me summarize two parts. So there are inhibitory, there are I demonstrated two different types of signals destabilizing synapses. One is inhibitory receptor, such as NOGO receptor 1, that generate protrusion backward, outward the synapse, but the extracellular adhesion molecule do somehow similar work. And in normal tissue, it will be really useful because they can scan more cells. But in tumor tissue, they can be good, they can be bad because they can be used as a tumor cell evasion, but it can also promote serial killing. And the last part, I will briefly talk about the actual 3D work. So we collaborated with Dr. Nurijan, who is expert in microfluidics, making 3D tissue chips. So very simple pipetting, we can make this collagen gel blocks embedded with the cells. So we implanted the HeLa cells and added NK cells and look at how they kill. So they migrate and engage and kill the tumor cells. So we can directly visualize it. And we did a simple experiment. Just we varied the concentration of collagen gel, and so that somehow mimicked the fibrosis going on in tumor tissue, only the tumor tissue. And what we found is actually there are less and less NK cells infiltrating into the tumor cells, and they kill tumor cells by contact-mediated mechanisms. So their killing was suppressed in high concentration collagen gel because their migration is restricted. But one interesting that they found is when they somehow get into the gel, they can kill cancer cells better, much faster killing for higher concentration of collagen gel. So in terms of collagen, we can simply change their concentration and do experiment, but changing concentration can change their microstructure as well as mechanical properties. So mechanical properties, actually Morgan Hughes did beautiful work using polyacrylamide gel with various stiffness and found actually the same kinds of things. So in stiff gel, the cancer cells are much higher cellular tension, and that make pore formation by purpuring much faster. So that's why. But we explored the structure, nanoscale structure by collagen. I will skip many things because of time. And we can also control their shape. So we can make it round or elongated, or we can make shape change and the topography change at the same time. So we make micro patterns of cells and observe their killing. And let me just show you we got some similar results. So we see the best killing with this elliptical shape with horizontal nanostructure, and that has something to do with, again, Roel-mediated cytoskeleton tension is the highest in here, and we see actually positive correlation between cytoskeletal tension and killing. So somehow that converge into the cellular tension. So this is just the summary of the second part. So ECM actually play many roles in NK cell cytotoxicity. So they regulate the biophysical property, regulate the state of tumor cells susceptible for NK cells. And they can also generate some physical barrier as we increase collagen concentration, less killing by limited migration. But at the same time, they can regulate the interaction mode, so facilitate detachment. That can be better for serial killing, but that can be bad in some substances. And the T cells are sharing some of the mechanisms, but not always. And based on what we observed, actually we have some tumor microenvironment and the metastasis, and the metastatic cancer cells tend to have elongated morphology and the lining migrating along the lined collagen fibers, or if they undergo extravasation, they are rounded shape. And I found actually NK cells are pretty good at controlling these, killing those two types of morphological different cancer cells, so that NK cell may not be really good for solid tumor treatment, but they may be really good at for controlling metastasis. Okay, so that's all I have for today, and thank you very much for your attention.